So I'm really pleased to be here with Lars and Frank from Michigan State University to talk about the 142-year-old Beale seed bank experiment. And I was just telling these two that when I read about this in the New York Times last summer, whenever that was, I was thinking as I was reading it, I was like, oh, I am definitely going to ask them to be part of the Stewardship Network's monthly webcast, whoever they are. This is such a cool project. And I get down reading the article, and I was like, oh, there's Lars, and there's Michigan State, and there's Frank. I'm like, oh, these people are our friends. So I was like, that's going to be even easier. That's not a cold ask. So as I said, I'm Lisa Brush with the Stewardship Network. I'm really pleased to be here to host the Stewardship Network's monthly webcast. I'm here with Lars and Frank from Michigan State University, where they are doing awesome work that is relevant to all of the kind of work that we all do in the Stewardship Network out in the field. And so really pleased to be able to share the research that they've been doing. As you all know, the Stewardship Network is a 501c3 with a mission to connect, equip, and mobilize people and organizations to care for land and water in their communities. And so we are the kind of backbone support for all of you, providing all kinds of tools and resources to enable money, knowledge, these kind of webcasts to enable you all to stay out in the field doing the work that you do to care for land and water in your community. So thank you for that work that you do. We are a national and international award-winning group that we started in 2004 as a nonprofit, 6 years after we had run as a project based in Southeast Michigan. We now work with people all across the country, and we have our 20 member communities, place-based networks. 11 years after we were started, we were called the up-and-coming conservation organization with the right trajectory. So it's really been a pleasure to be here working with all of you online and in the field doing the work that you do. We are all about partnerships and collaborations, and we work with all kinds of different groups. We have our place-based member communities, as I said, and those operate in specific geographies. And Rob, go ahead to the next slide. Let's see. As I said, those are locally place-based member communities, and they're made up of all of the organizations and individuals that care for land and water in their communities. And then one of the cool things we do is we link those place-based member communities together so that they can learn from each other. As an example, Peter, who's working with the Great Lakes Islands, year-round populated Great Lakes Islands, reached out to Dylan in Santa Cruz after we had connected them to share ideas and information and accelerate and advance the work that they do. So that's a wonderful part of the network, is the work that they do. This year is the 15th year of the Stewardship Network's conference. And many of you know that we're usually holding that on Michigan State University's campus, but we will be virtual this year. And the great thing that we were able to do, because it's a virtual event, we've always thought of our keynote and featured speakers as a cohort, and we've always wanted to invite them back, but logistically, that's going to be hard, and financially. And so this year we said, in honor of the 15th year, let's invite them back. So we have 24, 25 past keynote speakers. So it's a really great rock star lineup. It's taking place 2 weeks from today. It's in the afternoons of Wednesday, in the Eastern time zone, Wednesday, 26th, 27th, and 28th. And last year we learned a lot. We were able to have a really interactive, dynamic session where people said things like, I can't believe only you, the Stewardship Network, could replicate the in-person experience and make us feel so engaged and welcome online. So it's I encourage you all to check out the agenda, check out the conference webpage, and register and sign up, and look forward to seeing you in a couple weeks. I want to thank you all online who are financial contributors to the Stewardship Network. Your tax-deductible contributions help enable us to do things like these Stewardship Network monthly webcasts. So if you're new to them, these happen each and every second Wednesday of the month during the Eastern time zone's noon hour. You can put a recurring date in your calendar and go back to the website, check it out, and see if you're interested in attending. So again, I'm really pleased to be here today with Lars and Frank from Michigan State University. As I said, I'm Lisa with the Stewardship Network, and also really pleased to be here with all of you online. As I said, make liberal use of the chat box, asking your questions. Great to have you here from Ancaster, Ontario, and Arizona. Janine with the Burr Park Wet Meadow Project. Northern Ontario, Sleeping Bear, what a beautiful place. The village of Green Oaks. Manistee, Sudbury, Canada, Central Ohio. Great to have you with us from all over. So again, please make liberal use of the chat box. Looking forward to getting to know you all a little better online. And with that, I'm going to turn it over to Lars and Frank.
Thanks so much, Lisa. That was great introduction. And thank you everybody for joining. I see we have 79 participants. Oh my gosh, this is incredible. So let's see, maybe if Lisa wants to, or Rob wants to unshare their screen. Excellent. we can go ahead and share ours. All right. So thank you all for having us. We're really excited to be here. As Lisa mentioned, I'm Lars Brudvig. I'm a professor in the Department of Plant Biology at MSU. And maybe I can let Frank introduce himself.
Yeah, thank you, Lars and Lisa and everybody with the stewardship group. Thank you so much for inviting us to talk to you today. I'm Frank Tallusky. I am the professor and director emeritus. I just retired in October of the W.J. Beale Botanical Garden. I've been here on campus since 1993. And the Beale seed experiment has always been an interesting thing to me. I first learned about the Beale seed experiment as a graduate student in plant physiology. And so when the bottle was excavated in 1980, and the results published in the American Journal of Botany, it was a topic in one of our physiology seminars. And, I was like, wow, that's really cool. That's kind of neat. Wow, Michigan State's so far away from North Carolina, and who knows where I'm going to end up as a faculty member. Didn't even think about the reality of anything ever happening. And I didn't realize that in 1980 is when they changed the periodicity. I'll talk about that in a second, from a 10-year periodicity to a 20-year periodicity. So the next bottle wouldn't be excavated until 2020. And when you're in your early 20s or mid-late 20s, and you're talking about 20 years into the future, I mean, that's like, it blows your mind away. It didn't blow Professor Beale's mind away because he designed this experiment to really outlast his lifetime. And then subsequent people, and I'll mention this as well as we go through the presentation here this afternoon, excuse me, really pushed this thing much further than Beale imagined it could be, which is very exciting and a wonderful opportunity for all of us today in the 21st century. Now, this is something that started out in the 19th century. Seed germination has always been of interest to me, not so much as an experimentalist, as a researcher, but as a person who's a botanist who grows plants. I was always interested in how long can I keep my pine tree seeds in the refrigerator and keep them viable. One of the things that my first faculty position was at the University of Arizona in Tucson. And one of the things I became very fascinated with was the desert blooms. How long do those seeds remain viable in the soil? And then after a really good rainy year, it could be 20 years since the last major bloom, all of these flowers just come up out of the seed bank. And that was really fascinating to me. Just on being aware of seeds can remain in the soil in the seed bank undisturbed and ungerminated for decades. And when I moved to Michigan State, I had a whole bunch of conifer seeds with me. One of the seeds I had from back when I was a high school student doing science fair projects in the mid 1970s was an ounce of sequoia seeds, Sequoia dendron gigantea. And I went to our nursery manager at the time, Tim Boland, and said, hey, see if any of these germinate. This is 1993. I had these since 1975. And he puts them out, and lo and behold, these things germinated. I've been keeping in refrigeration, but it's 20 years later, these things are still viable. So seed germination has always been of interest to me. And so Lars, will set me up on the first slide. Really important that we talk a little bit about Professor Beale. Professor Beale started this experiment. He was a native-born Michigander. He was born outside of Adrian on the farm country. His family, his mom and dad, moved to Michigan, the frontier at the time, from upstate New York. And Professor Beale, the young William James Beal, spent a fair amount of time here in the state growing up and then going to the University of Michigan for his AB degree. And then went on to do some teaching back in New York, upstate New York, and eventually went on to Harvard University and became a student of Asa Gray's and Louis Agassiz, very well-known scientists. Of course, Asa Gray is a very well-known botanist, and so had some very good classical training. And after a short period of time after graduation, he eventually came to Michigan State and lectured in July of 1870 before taking a full-time position as the professor of botany and horticulture at Michigan State, starting in 1871. And he eventually got an honorary degree from the University of Michigan, an honorary PhD. But again, in those days, having a PhD wasn't a prerequisite to be a faculty member, professor. But anyway, what was interesting is that he was a professor of botany and horticulture. The horticulture department got spun off in its own independent department, and he became the professor of botany and forestry. So he really started forestry in the state of Michigan. And that's another lecture. If you ever want to lecture about forestry in the state of Michigan and growing trees and how that all got started, I can give you a talk on that someday. But to kind of make the longest story a little bit shorter, in 1879, he started the Beale seed experiment. And main thing that he was interested in with the Beale seed experiment, again, next slide, was to try to find out how long do seeds remain viable in the soil. And although I don't have any written information for this, the one thing that's important to remember is that Michigan State was the first land grant institution, so granted by the US government, the federal government, and was very much involved with helping farmers learn how to grow crops on this new lands of the frontier and around the country and get the best yield, best productivity from the land. And so Professor Beale became involved with an aspect here in the state of Michigan called the Farmers Institute, where he'd go to county, kind of like an extension office today, and present lectures and take questions from farmers about how to help them increase their productivity. And at that time, the state of Michigan saw a lot of immigration, and a lot of people coming in from various parts of Europe and from the eastern United States. And they were buying a lot of cut over land that was cut over by the timber companies and sold off, because the timber resources were kind of the first natural resources of the state to really be exploited. And how do you grow crops up in Grayling on 300 feet of glacial sand? Maybe you're better off just growing trees on that glacial sand, as we found out 100 years later. But anyway, so the question would come up is, in those days, we didn't have herbicides, we didn't have John Deere tractors. How did you control weeds? Well, you got a hoe and you went out there in the hot summer sun and you had a long row to hoe. That old saying, you got a lot of work ahead of you, you got a long row to hoe. And you mechanically yank these weeds, pull these weeds out. And the farmers would be probably asking, how many times do I have to pull these weeds out before there's no more weed seed in the bank and soil? So, with that, probably that in mind, and other aspects of just a pure scientific curiosity of how long do seeds remain viable, Professor designed this experiment, and he designed it in such a way that it would go long past his lifetime. I can't imagine he didn't think about that when he designed it, because he started out with 20 bottles with 20 seeds, 20 species of seed in each bottle, with 50 seeds per species. I have 21 because, as we'll see here in a little bit, there's a little confusion with verbascum, and we'll talk about that in a little bit. So 21 different seed species per bottle. He also had some acorns and walnuts, oak and juglans that he buried next to the bottles, but those seeds did not remain viable for a very long period of time. We'll kind of address that as well. Verbascum blattaria, however, the verbascum in general has the longest germination record of the plants that he planted, and the most viable. I mean, the greatest germination percent. So in the late 1998, we adopted the verbascum flower as our logo for the Beale Botanical Garden. We thought, the longevity of the garden, the longevity of seeds made an appropriate application to use that flower as our logo. And you can see a picture of me there, much younger, 20 years ago, in amongst the verbascums blooming in 2001. Next slide. So as I mentioned before, this is Beale's own words. You can read that. I'm not going to read it in its entirety, but it basically describes how he set up the bottles, the 20 bottles with 20 different seeds, and buried them in the soil. And if we have next slide, we can see how he arranged those bottles. They were buried 18 inches below the surface of the soil, someplace in a secret location here on campus. And Lars will talk a little bit about that and his aspect of it. But you can see that he put the bottles in at an angle, upside down, left the bottles open. So he mixed the seeds with sand that he excavated from this uncultivated region of campus, had never been cultivated, dug down several feet, got the sand, mixed it with the soil, mixed it with the seeds, and then filled up each one of the bottles to have an exact replication of the number of species and number of seeds per bottle. And he left them uncapped because he wanted to allow for gas diffusion, oxygen and carbon dioxide to move in and out of the bottle, moisture to move in and out of the bottle. He didn't have them vertical for obvious reasons. He didn't want them to fill up with water and become flooded and kill the seeds. So this is how the bottles actually appear below ground. And we went out to excavate them in 2000 with Dr. Zavart, and then this time with my team. This is basically what we would see. Of course, diminishing number of bottles. We're only down to 4 bottles. Okay, next slide. And this is what I was talking about. So Quercus rubra, which later was thought maybe was Volutina, which was later described as rubra and Juglans nigra, the black walnut, were buried in there. But really, nothing was observed with those seeds since 1905. So it's a neat afterthought. Yes, he did try tree seeds, but most of the seeds that he worked with were in the bottle and were primarily weedy species. Not exclusively weedy species, but pretty much fairly common weedy species. Next slide. So here's how the experiment was set up. Professor Beal buried the bottles 1879. One of the unfortunate things that you'll see in a second here is he never did a 0 time, a baseline germination record. So we have no idea what viability of the seeds were going into the study until the first extraction of the bottle in 1884. And that's the first data set we have for germination for all the species. And that becomes important, as we'll see in a little bit. So he did the last bottle he extracted here on campus was in 1909. When he retired in 1910, passed the experiment on to Professor Darlington, who also was a botanist in the botany department and also was the director of the herbarium at MSU, as was Professor Beal. And Professor Darlington excavated dutifully a bottle in 1914. And in 1919, he was going to do an excavation of the bottle, but unfortunately the ground had already frozen. And so we had to postpone the excavation of the bottle until 1920. And there was some discussion we had because of the pandemic, and as you'll hear from Lars, how the pandemic impacted us. Did the pandemic, the Spanish flu pandemic, impact the excavation of the bottle? Wouldn't that have been an irony? But I mean, it's 100 years ago that we had the Spanish flu pandemic. We don't see anything in the records that was in fact the case, but we do know it was delayed. And so hence, ever since 1920, we have done the excavation. The excavation's been conducted in the spring of that year rather than in the fall of the previous year. And that's where we move it. And so the experiment was continuing on a 5-year basis. But in 1920, Professor Darlington noticed that, we're pretty much getting the same seeds germinating. Let's extend this to every 10 years. And let's really stretch this experiment out so that, we'll take full advantage of Beal's design. And that continued on. Professor Darlington in 1960 added a colleague to the excavation before his retirement, and passing it on to Professor Bandersky. And Professor Bandersky had his colleague, Dr. Kivalon, do the next 2 excavations in 1970 and 1980. And this 1981 is the one that's seminal to me. And it's really interesting to talk to the team. Lars might want to reflect on some of this as well. How many of my colleagues who just joined the team, when they were graduate students or undergraduate students, heard about the Beal experiment when I did the excavation with Dr. Zavart. So it's a neat story, not only from the science aspect of it, from the social, scientific, collegial, professional aspect of it too. I mean, it's a really neat program. But in 1980, Dr. Bandersky presented to the faculty of the botany department and a discussion led by the department chair at the time, Gusta Zuten, and should we extend this period again to every 20 years? Because again, verbascum seems to be the one thing that's germinating consistently, and everything else seems to not really be germinating all that well, or not at all. And so it was decided, yes, we'll postpone the next dig until the year 2000. And I came here, and like I said, in 93. In 99, Dr. Zavart came up to me and said, Frank, I'm not going to be here forever. Would you like to be involved and take up the reins of the Beal seed experiment? How do you say no to that opportunity? I mean, I mean, it was a great honor, and it was coming full circle from 1980 for me, and I was honored to be part of that experiment. And so we dutifully excavated the bottle on that fine April morning in 2000, and you saw that picture of me amongst the germinated verbascum. Fast forward now to the second decade of the 21st century. I was thinking about the experiment a couple of years ago before we conducted it, and I approached David Lowry and said, David, how would you like to be involved with this study? And I said, David, I'm getting up there in years. There's no guarantee that I'm going to be around for much longer. I hate to see the experiment not have continuity. And as Dave reminded me a little bit later, it wasn't until a few months later that I actually suffered a mild stroke. And when David got the word that I had suffered a stroke, it was like, oh my God, thank God I more or less recovered, more than less. But I didn't realize until talking to David how much that impacted him. It's like, wow. And we decided that we really need to increase the diversity of the team in terms of scientific expertise. And so we approached Lars, who obviously has interest in conservation, restoration, ecology, Marjorie Weber, who's a evolutionary scientist, and a postdoc who's working with Margaret Fleming, who very interestingly actually worked with the USDA Seed Lab in Fort Collins. And so the team was expanded, and the new team was born and passed it on to the next generation. Next slide, please. So what happened in the study? We can see this is a copy of the data table that I had published with Dr. Zavart in 2001 in American Journal of Botany, and we can see all the things that did and didn't germinate over time. Let's take a quick look at one of the things that we are going to discuss here and focus on, and that's verbascum. And so if we look at verbascum thapsus, Beale reported that he had seeds of verbascum thapsus. And we can see we had a really great germination, 84%, that's 42 seeds germinated in 1884. It's one of the more robust germinations. As you can see, there's a couple others. We had 100% germination for one of the brassicas up there. But anyway, pretty good germination. Wasn't sure about germination in 89. Not sure what happened with that study. He was just recording pluses and minuses. We didn't actually have hard number data, unfortunately. Beale didn't report that. He went back to hard data again in 94, and we can see 36% and then 32%. But then nothing seemed to germinate in 04 and 09, 1904, 1909. He had a plus germination reported for 14, and that would have been Darlington, but Darlington didn't see any germination again in 1920. Oddly enough, in 1930, Dr. I hate when my brain does that. I must be getting older. But anyway,
Darlington.
What he noticed is that we had a 62% germination, but the plants were not verbascum thapsus. They were verbascum blattaria. Well, the seeds of verbascum in general are very, very tiny. And it's really hard to tell the difference between verbascum blattaria and verbascum thapsus. So we have no indication in the literature of why this switch may have taken place. I can't imagine that Dr. Beale would have mistaken a plant of thapsus for blattaria, given what an outstanding botanist he was. It's possible that they ran out of thapsus seeds and they replaced blattaria seeds, but I think Beale would have made a note of that. Maybe it was a student who was collecting the seeds, and maybe they just got the seed batches confused. Anyway, something happened, but we know that we have now in the study pretty much verbascum blattaria, and you can see very well represented, 62%, 68%, 74%, 70%, 42% in 1980, and 46% in the experiment I was involved with. But we also noticed that there were two species of verbascum that didn't look like blattaria, but they didn't look like thapsus, and that's why I added that verbascum spa to that list there. We had two germinants of that. So really, we had a 50% germination on verbascum species in 2000. which was phenomenal. The other thing that was interesting was that we also had a germination of Malva rotundifolia, which had a name change. And so we can highlight that. We can see that Malva has been very inconsistent. It germinated in 84, no germination, then 6% in 99. And then nothing shows up again until 1980, and then for us in 2000. So what this tells us is that clearly Malva can have a very long germination record, just like verbascum does. But why are we seeing this punctuation? And this is actually answered if you go back into the original publications of Professor Beal. And Professor Beal was a very keen observer. He always emphasized to his students, keep squinting, keep looking, keep observing. And one of the things that he did as a good observer was he noticed that he saw the seeds, the seed coats of Malva in the sandy soil mix, but when he broke them open, they were hollow. And so those seeds weren't viable. And so when they counted the seeds out, they thought they were counting out viable seeds, but they had a very low viability, very low seed set in those seeds. A lot of them were hollow. They were just hollow husks. And this again goes back to my statement earlier. I said, we don't have a T0 germination record. And if we would have had a T0 germination record, Professor Beal would have known right from the get-go, before the experiment even started, that, hey, these seeds have a low viability rate. So anyway, very interesting data. Next slide, please. So comes the year 2020, and we're getting ready to excavate the next bottle. And we have the team, I have the team assembled, seed botanist assembled. There we are, ready to go. Lars, take the story away.
Thanks, Frank. That was great history right there. And so like Frank said, this brings us up to just before 2020. And so Frank had assembled this team. You probably noticed that we have more team members now than we've ever had in the past. And one of the main reasons for that is that we have been realizing more opportunities and more applications than were ever envisioned by Beal or past stewards of this experiment over the course of its history. I first heard about the Beal seed experiment when I was in graduate school as well, but I was in graduate school for a really different thing from Frank, and that was restoration ecology. And I was learning about seed banks because of their application and relevance to restoring damaged ecosystems, and also in conservation biology, where the seed bank can be a friend or it can be a foe. It can be a friend because perhaps sometimes seeds of plants that we are trying to promote in conservation and restoration are in the seed bank and may bring, happy surprises once we start a project, once we reintroduce fire or reinstate hydrology by breaking tile drains or something like that. We might get some of the target species that we're hoping to promote. But in other cases, we get challenging weeds, even invasive species living in the seed bank. So seed bank is highly relevant to restoration ecology. An interest of mine, real honored to be involved with the project. We've also, we have questions that have emerged about how evolutionary change happens over time. And there's opportunities to look at things like the genome of these 142-year-old seeds compared to the genome of seeds collected from the same species today on our landscape. Bringing the Beal seed experiment into the genomics era, of course, nowhere on Beal's radar at the initiation of the project. And Marjorie Weber, David Lowery bring expertise that bears on that front. And like Frank mentioned, Margaret Fleming is a seed biologist. And I'll talk about this a little bit later, but is very interested in this idea of zombie seeds. So seeds that are still alive. Seeds are living things, right? But don't quite have the machinery working anymore to be able to germinate and develop into new plants. And that might be apparent when looking at not the genome, but the transcriptome of these seeds. I'll talk about this a little bit more. So really, really cool new applications and ideas that are floating around in our team. We also need to acknowledge the MSU communications team. So this has been a new sort of experience for me, doing science, which has really captured the public imagination. And Kim Ward, Kevin Epling, and Derek Turner through the MSU communications group have been critical for helping get that message out, connecting us with journalists, helping to write news articles, taking wonderful photos. You'll see most of the photos here are by Derek and so on. So I should back up. So this brings us up to 2018 or so. 2019, Frank had assembled this new team. An interesting aspect of this is because of this 20-year time step, we essentially need to do the experiment and recruit new team members every single time because we're doing and handing off. I'll be 60 years old at the next dig, and it'll be time to hand the experiment off to the next stewards of the Beale seed experiment. All did not go quite according to plan for probably obvious reasons. The 2020 dig should have happened in April of 2020, and I'm sure we're not going to forget for a long time probably what life was like in March and April of 2020. The pandemic was just setting in here. There was a lot of uncertainty. We didn't know if we were going to be able to access our facilities on campus, if we would even be able to get in and water our plants. The last thing we wanted was to unearth a bottle, put everything in motion, and then just have it collapse. So we made what in hindsight was a very smart decision, and we delayed for a year. So the dig happened in April last year, 2021. It happened under the cloak of darkness, which I'll get to in a little bit, and was kicked off by an email Frank sent around to the group. So we were just so excited. We've been planning for this for over 20 years, and for years, very closely as team members. Frank sent around some lyrics, borrowing a little bit from a couple of famous songwriters, but adopted for the purposes with the Beale seed experiment. We met up on campus at, I think it was 3:30 in the morning. So why did we do that? Why did it have to happen under the cloak of darkness? Well, there's really two reasons for this. The first has to do with the interest that this experiment has generated. There's almost 100 of you on this webcast today, which is amazing to see. And there's been so much interest in the Beale seed experiment. We want to see it continue on. And the last thing we'd want to see was somebody get a little bit too curious and want to go searching around on campus. So we want to keep that location secret to just the team members and folks on campus who, make plans about like where buildings go and pipelines go and stuff like that on campus. So the folks that need to know. The second reason is a biological reason though, and that has to do with triggers of seed germination. There's a number of these, but a really important one is exposure to light. After seeds have been underground in the seed bank, exposure to light can start that machinery and process that can lead to seed germination. As Frank mentioned, these bottles are all buried underground in a row. And so this year we were digging up, or last year we were digging up the 2021 bottle, but there were also the 4 remaining bottles underground. And so we did not want to dig during the day and have those be exposed to sunlight and accidentally start germination in the 2040 bottle or the 2060 bottle and muck things up for the future. So we met up on campus. The location is, again, it's a secret, but we have with that a secret map. I should tell you, when we were getting ready for this, I mentioned the secret map to my daughter, who immediately went down the stairs and started rummaging in our basement in a trunk that we have. She was convinced that the treasure map was in our basement in this blue trunk. Of course, it was not, but it tells us how to find the location of the bottles by triangulating from certain landmarks on campus. Things like, trees on campus. So we did this triangulation. We dug, you can see that it's just like turf grass here. We remove the grass, we dig down about 1.5 feet, very carefully at the end. The last thing we want is to hear the sound of breaking glass at the base of our shovel. So we start off with this larger spade, we get down to a hand trowel at the end. After some amount of time of digging in the middle of the night, it was cold, it actually snowed a little bit on us. Success, we found the bottle. Here it is. Brought it for a visual aid today. This is the same bottle that we dug up last year. It's the same style of bottle. Remember, upside down like this in the ground. It was packed with those seeds with sand. You can see it here. This is Marjorie Weber's hand on the left. Marjorie was the one who found the bottle. Here's Frank adoringly gazing into the bottle soon thereafter. So we got this bottle, and now it's off to the lab. So the next steps are, we have a tray, like a greenhouse flat with sterile potting soil. We spread this sand seed mixture onto the surface of it. We all got to take turns doing this. Part of the team. Here you can see Frank taking his turn. And then this goes into a growth chamber. So this is a controlled environment where we can do our germination trial. It gets watered. And so that exposure to moisture is another important trigger of germination. It's getting light here. It's getting, the sort of atmospheric conditions available in the air. And this is enough to trigger germination for many species of plant seeds. And now we wait. But we did not have to wait long, because lo and behold, only 8 days later, we saw the first germinate pop up. And that was the start of something. So in some, we had 20 seedlings emerge out of this 2021 bottle. But what species are they? So we have to grow these. They're getting bigger. As they get big enough to replant, we put them into pots, and then we continue growing them so we can identify them. We knew at this point that we were working with verbascum. That was clear to us. But remember, which species of verbascum? Well, there are ways that we can probably tell vegetatively just by looking at the leaves. They do differ in terms of like their texture, for example. We wanted to be sure. And the way that you can really be sure is in the flowers. Moth mullein has really big showy flowers like Frank showed before. And you've probably, many of you seen common mullein that has also yellow flowers, but much smaller. So we're growing these in pots. Verbascum is a biennial. And so a trigger of flowering for it is that it goes through a winter. And so we did this artificially by sticking these pots into a refrigerated room for about 2 months to simulate a winter. That triggers biology for these plants. And then this is where we're at right now. So these are our plants. I just took this picture on Monday. You can see that we have flowering for one of the plants so far, and we're waiting on the rest to confirm that identification of Verbascum blattaria. Along the way, we did a couple of additional trials. We ran the whole experiment exactly, or as close to exactly as possible, how it was done in the past. Once we got to the end, we thought, let's have fun. Okay, and so we wanted to try out a couple of additional treatments that we know can stimulate germination for plants. One is the application of liquid smoke. This can be important for seeds of a number of plant species that are adapted to fire-frequented environments. One of the species, just out of curiosity, that was in the bottles originally and has never germinated is Erectites, Hyracefolia, common name fireweed. So there's no evidence in the literature that has smoke-induced germination, but we didn't know. So we thought, let's do a liquid smoke treatment. We also did a gibberellin treatment, which is a plant hormone that can stimulate germination as well. So we did this. What did we learn? No new germinants, but we tried and had a little fun on the way. And that was a bit more answer for us, that was not part of the puzzle, at least at this point in the history of the experiment. Okay, so what's this year 141 bottle taught us? Well, first of all, Verbascum continues to germinate at a high rate. These seeds were last touched by Professor Beal 141 years ago, and they've been sitting in these bottles ever since, alive, and not only alive, but able to germinate when they get the right cues. We saw 40% germination of these 141-year-old seeds, which is quite similar when we look across the rates that we've seen for Verbascum in history, particularly compared to the 100th and the 120th year bottles, where there's been 44 and 50% germination respectively. We continue to help answer Beal's original question, which is a simple but fascinating one. How long can seeds remain viable in the soil? This is still relevant to its original motivation of farm weeds. Farmers still have weeds in their fields, but the relevance has only grown as we've developed these fields of conservation biology, restoration ecology, and other fields within ecology since Professor Beal's time. And I think it's amazing how this just demonstrates this value of long-term research. I mean, what a vision and what a effort for nearly a century and a half now to continue this experiment on, pass it on from generation to generation of plant biologists. And it's been just such an honor to be one of those stewards of this experiment. It's also been incredible for communicating science. So I think Lisa mentioned that she first heard about this through a New York Times article. There are actually 2 articles written in the New York Times about this experiment. There was an article and a podcast done through NPR and a number of other news stories, in each of which we got to talk about this amazing experiment, but also, the science of seed biology and present ourselves as scientists to the public. So let's look ahead a little bit. What's the next step for the Beale seed experiment? Well, first of all, we have to wrap up the year 141 trial. We need to finish identifying these verbascums to species. And then at that point, we'll be done with the experiment for last year. We'll write up the results as we've done in the past, and we'll publish them in a scientific journal. 4 bottles are still underground at the secret location on campus. The plan at this point is still to unearth 1 bottle every 20 years. So the next one, we will get back onto the normal schedule, which of course was disrupted by the pandemic, but we'll go back onto the planned schedule to get the next bottle in 2040. As things stand right now, maybe decisions will get made down the road after my career is over, perhaps, but at least at this point, the last bottle will come out of the ground at 2100, 20th bottle. At the same time, we are talking about what is next beyond or alongside what we are calling now Beale 1.0, the current Beale experiment. So what could Beale 2.0 look like? It's been this amazing experiment on campus. Let's keep Michigan State at kind of that centerpiece of seed germination research like this. But what would it look like? Well, and what are the interesting questions? How can we set up an experiment that would be flexible into the future to be able to answer new questions that we can't even maybe imagine asking right now? Well, one thing that we have decided is that we should set it up as a paired bottle experiment. So right now we unearth 1 bottle every 20 years at this point. And that bottle is dedicated to the germination trial. We still want to do that. But what we would love to have right now is a second bottle that Margaret, for example, could grind up seeds in and ask, do we have these zombie seeds? Seeds that won't germinate, that don't germinate, but are still living there, with working genetic machinery, just not quite able to germinate anymore. Or grind them up and do a genomics test to evaluate old seeds against present day seeds, or again, who knows what other sorts of studies could be done that would have to be destructive by nature. We're interested in including some of the same species as Beale 1.0. The one thing that seems clear is we need verbascum. Verbascum is the heart and soul of the Beale seed experiment. We need verbascum. What other ones as well? But we also are interested in adding some new species. For example, some species that are of high conservation interest, where we might want to know about longevity in the seed bank because of their applications to conservation of threatened and endangered species or to the restoration of imperiled ecosystems. So the vision right now is to have kind of a mix, a similar sort of bottle design with duplicate bottles that'll get unearthed at each time step. Some of these weedy farm sort of species, but also some native conservation interest species. And we've also started conversations with others. We're excited about the idea of building this into a network, preaching to the choir here, right? But this has been such a neat experiment, and others are interested in the same sort of thing. Right now, we know a lot about seed longevity in the seed bank for these particular species at this one wonderful, but one location, Michigan State's campus. It has the environment that it has here. How would things look, though, if we went to Ohio State's campus, or the University of Florida, or somewhere else in the world, and they did the same process, the same experiment, and we could all share our results and ask about those sort of context dependencies. That's something that's really exciting to us right now, is building this with, of course, Michigan State as a center point, but as a network. Okay, so if you would like to read more, here are some resources. I think I have this right. I will drop these into the chat for you so that you can link to them as well. But these are informational resources about the Beale seed experiment, a wonderful article that Kim Ward wrote up for us at MSU Today, the New York Times article, and the NPR article, which I had referenced before, if you're curious to hear a little bit more. And with that, we can say thanks. Hopefully we have some time for questions. We'd love to hear from you.
Great. Yeah, what a wonderful, wonderful project. And, you know, when I first read about it, I was thinking, Lars, about the restoration pieces, because, you know, all of you online, many of you probably know the stories of the plant that hadn't been seen in 60 years, and then you start restoration and boy, it shows up, you know, and so neat. And then I love the fact of the questions that hadn't even been thought of, right, 142 years ago. And now we can ask those, and what will they be in the future? So what a wonderful, thank you so much for sharing this awesome project with us. Couple different questions. So, and then Frank's been answering some of these online, but I want to make sure that we answer them verbally for folks as well. So yes, go ahead,
I was going
Frank.
to say real quickly, there's a really good one about the current study, and are we doing anything with the current seeds? And I was typing, so I wasn't listening to Lars' explanation quite as much. But I did save F1 seeds, both closed germination and open germination of verbascum in 2001. And I also kept subsequent F2, F3, F4, F5 generations. We planted the plants that germinated in 2000 into the Beale Garden, and we have kind of a continuously running population of those plants in the Beale Garden since 2000. And so it will be interesting. One of the things we talked about was comparing the genetics from the seeds that germinated this year to the F1s and all the way up to the F20 population, and then looking at the broader population of verbascum in the state and where these would have originated from, and see what may have changed over the course of 140 years of evolution. So I mean, has the plant evolved much, basically, in the past 140 years?
Great, Frank. And that actually alludes to one of the questions that Matt asked, that you seem to imply the answer here. What do you do with the species that do germinate? So maybe tell us a little bit about the Beale Garden. Okay, so
I thought it'd be very interesting to put these plants on display down in the garden and kind of use it as an outreach educational moment. One of the things, unfortunately, is we also ended up inadvertently contaminating the gene pool locally within the garden for any verbascums that might have germinated, plantaries that would have germinated in the garden. So, it's kind of, you take your chances. But we put those plants out in the garden for demonstration. And then my key thing was, then from a scientific value, not even knowing what would be possible, because even 20 years ago, sequencing a genome was horrendously expensive. We could do it. And a human genome had, I think, just been published or was in the process of being published about 2000. But I mean, it was such an undertaking. And now it's really relatively inexpensive to sequence. So, but again, trying to keep that Beale state of mind of thinking forward and planning for the future. That's why I collected those seeds from the different generations year after year.
Great, thanks. A couple other questions that folks asked. Steve asked, do you wait longer to see if anything else does germinate that take a little bit longer? And Frank, do you want to say a little bit about the answer that you gave in the chat to that?
Sure, I think Lars might be able to address that a little bit further. I don't remember exactly, was it 6 weeks or 8 weeks? How long did there was a, so we extracted the seeds mix from the bottle. We spread it out and put it in the growth chamber for several weeks. And it was, Lars, was it 8 weeks?
Yeah, I think it was about that. We waited, I think it was 4 weeks after the last germinate came up to make sure that it didn't look like anything else was going to come up. And then we stuck the flat into the refrigerator. For some species, that second cold is important for stimulating their germination. And we got, I think, 4 more germinants after that. all verbascum again. Interestingly, it was after that second cold that Malva came up in the 2000 study, wasn't it, Frank? If I remember
Yes, that's
right.
correct.
So perhaps that was important for Malva. And then we again allowed germination to happen over the course of a few weeks. And then we waited, I think it was 4 weeks again, and after the last germinant had come up. Before at that point, we decided, okay, we followed the protocols that have happened before. And it was only at that point that we tried the liquid smoke and the gibberellin to see, just out of our own curiosity, if that mattered at that point. It didn't. So at that point, it ran for another, I think, month almost with no germination.
And again, if you think about it, gibberellic acid was completely unknown in 1879, 1884. Gibberellic acid, I think, was only discovered in the 1940s or 1950s. And the same thing with the effect of smoke on germination. Who knew that there would be species that would be fire dependent back in the 19th and early 20th century. We do have one plant that never germinated in the seed experiment called fireweed. Lars looked up fireweed and found out that it really isn't smoke dependent, but we thought, well, maybe these
Well, not for some reason, right?
and fingers were crossed that some of those seeds would have germinated, but they didn't. And I was really kind of hoping that a Malva seed would have germinated, but again, given the low percentage of seed set that the experiment started with, the odds were definitely stacked against us. I think we were lucky to see it in 1980 and 2000.
Yeah.
And maybe we'll see it again in 2040.
I know, right? Yeah.
Hopefully I'll be here to see it.
Yeah. So I was thinking about the 2100. I was doing math. I'm like, oh, I don't think I'll be around for that one. So a couple other comments and thoughts. As Barb says, for 2.0, I'd love to see indicator species from our rare plant communities. This might inform our restoration efforts for those communities.
Totally agree. Yeah. And the seed bank it's fascinating, and it's really hard to know about too, right? Like we do these restoration projects and things show up, and a lot of times we infer it's probably from the seed bank, right? Like, because where else would they have come from? Well, I guess it could have dispersed, or it could have been a tiny little plant that was just very stunted and suppressed that we overlooked. It's hard to know about, and I think that's part of the real value of the Beale seed experiment, because it is an experiment and we can know for certain that these seeds are living for exactly how long they're living for in the soil and still able to germinate.
A couple things I wanted to add to that. When we did the experiment in 2000 and we got a lot of press, it seems to really get a lot of public relations when this happens. I'm glad to see that the public and the media are interested in the story, because again, it gets people thinking about science, conservation, and so forth. But I got a phone call from a farmer down in the southwest corner of Michigan, and his family had been farming that land since the late 19th century. And of course, one of the first areas to be farmed in Michigan were the old prairie, the prairies remnants in that part of the southwest corner of Michigan. And there's a lot of extirpated species in the state that are from that kind of that particular habitat. But he said that they dug a farm pond and they spread the spoils out in a pile near the pond, and lo and behold, all these prairie species started germinating.
Cool. And
unfortunately, I never followed up in terms of what species I probably should have documented that. Again, it's kind of an incidental thing because it wasn't possible that they blew in from someplace. What was the nearest population? Were they really seeds that were over 100 years old? But there are so many stories. There are stories from Israel about date palms being excavated by archaeologists. There's the classic story that for a lotus seeds from a dried lake in China. And actually, Jane Shen Miller, who was the scientist that worked with those seeds that were carbon dated to be, I think it was over 2,000 years old, as a professor at UCLA. She's actually a graduate of Michigan State University. And when we did this study, she contacted me via email a couple of months ago and gave us one of the F1 seed generations of the seeds that she collected in China that germinated. That was a story I think was reported sometime in the 1980s or such. I don't remember exactly when that was published, but there's stories about buried seeds in permafrost that seem to germinate. So this is something, seed banks and the viability is well known. There's a story I heard, and I can't confirm it, but it's interesting if it's true, about a species of plant in the Mississippi Valley in St. Louis area that was at least extirpated, if not more or less extinct in the wild. And when they did a dredging of the river, and they put the dredge spiles out, all of a sudden this thing started germinating. So this is from a restoration ecology standpoint, from a restoration of threatened and endangered species, of reduced genomic population, breeding size populations for plants. I'm sure that zoologists and zookeepers would be absolutely wishing that this would happen for animals. I think we're really fortunate that this happens for plants. There was one question about verbascum, being invasive. The flip side of that coin is, if you've got a species that's invasive, if its seeds are going to be viable for over 100 years and they get into the seed bank, how are you going to control that in the environment? If something that is an invasive species, but it has a very low longevity in the seed bank, and I think that might be true of what's the invasive, the garlic mustard. If it's true of garlic mustard, then an eradication program, a control program for garlic mustard may actually function better than one for verbascum.
Right, as somebody mentioned in the chat. All right, so we're coming down to our closing final minutes here. I've got a couple other questions that I want to ask you. One is about, as Bob says, any significant imitator experiments been launched in the last 142 years? And Lars, as you mentioned, kind of starting to create a network of this, but has there been anything like that? Then I also want to ask another question that Peter poses of, is it worth analyzing the protein, amino acid, and fatty acid profiles of the seeds over time for clues about internal support for long seed survival?
Yeah, that second point is something that Margaret has talked about, Margaret Fleming. And that's a great question. That's exactly the sort of thing that Margaret thinks about all the time. There have been other buried seed experiments, none anywhere near as long as the Beale buried seed experiment, but in other systems to try to understand the same sort of questions. How long can these seeds stay viable and able to germinate in the soil? And again, people care about that for the things that are of conservation interest, but also about the things like Frank mentions that are going to be hindrances to our conservation and restoration success, weedy, invasive sort of things.
There was a study that was brought to my attention that was started at Ohio University in Athens, which happens to be my master's degree alma mater. And I think it was back in the middle 20th century, where a professor there, inspired by Beale, set up a Beale type experiment on the campus of Ohio University. But unfortunately, they went and built a building on top of where the seeds were buried. This is something that again, I didn't catch hold of Lars's talk, but our IPF, our folks who are in charge of campus utilities and construction, they know where the bottles are, and so they know not to do anything there. There was one time I was walking across campus and they were doing some utility work, and I kind of walked up to the head person and said, "Do you know what you're doing here?" And he pulls out the map and he showed me on the map. He says, "Are you talking about this?" There was the Beale experiment. Thank you. Yes. Okay, good. Not to disturb that. So I said, yeah, no. And the fact that it's protected by concrete. I mean, as Lars described, there's this big, what is it, like 8-inch thick poured concrete wall around it. If somebody went through there with a trencher, they're going to hit that concrete wall before they even come anywhere near the bottles. And hopefully they would stop and go, "Whoops, what's this?"
Right. Great. Well, I know that Frank, Lars, you and I and the online could continue to talk about this for a long time. Maybe the thing to do is tune into the conference next week, and we'll pop into a networking room and continue the conversation online. I've dropped the conference link, so go ahead and learn more about that. Would love to see you all with us in 2 weeks. And then Sylvan, for those who don't know, these webcasts are recorded, so you will find them online at the Stewardship Network's website. You can look through the archives of all kinds of different ones, and we will be back with you during the Eastern time. So much like not quite like Beale's, but we've been doing this for like 20 plus years. And so back when webcasts weren't a thing that anybody did, and we didn't do this all the time, you will find the Stewardship Network on each and every second Wednesday of the Eastern time zone's noon hour doing these monthly webcasts. We will continue to carry them on. Lars and Frank, thank you so much. And what a great honor to learn about the tradition of carrying on the Beale Seed Bank experiment. And thank you
Thanks
all
a lot for having us.
Yes, really, really
Yeah.
appreciate it. And
Thank you for your
taking the
interest
time.
and thank you for having us. And I think this is information that's going to be useful across a number of professions.
Yeah, it is. And that's such a neat thing about expanding the team, right? So many questions to ask. Well, again, thank you all online. Looking forward to being with you next month and maybe in a couple weeks at the virtual conference. Take care and keep staying safe.
Thank you. Yep. Stay healthy, everybody.
Exactly.
Bye.